Hi. I was asked to explain Okazaki fragments a little better. So I'm going to go ahead and go over a couple things, and hopefully it explains more detail for other people who may have questions and I just can't answer them. So first things first, why are we doing DNA replication? Remember mitosis, mitosich. I scratch them, I need to repair them, or I need to replace worn off skin or sloughed off skin. Anyway, we want to repair, replace, or reproduce. So we go through mitosis, and during that we do DNA replication. So the ultimate goal is to make two identical copies of DNA. Okay? All right, what I want you to know is some enzymes that are really important in doing this, and I didn't get a chance to point out. You may have picked up as you were doing the work, but I'm sort of going to go in order, I guess you could say, is how maybe you might think they are in use. So it might help you a little bit. All right, so first one is, this marker is much better. You have topoisomerase. This is what we're using specifically is called gyrase, and it's to relieve the tension so you have your DNA strand. Remember, this is the unwound version, and we're talking about DNA that is super coiled. So we have to really kind of relieve the tension. We're not completely unwound, but as you can see, we get to that point where we're no longer in this. We kind of look like that. Okay, sorry, that's as good as I can get. Okay, so we kind of get to a point where we're uncoiled a little more. We're not super coiled, as it's called. Okay, we also have helicase. Again, I think I said before to you, it's a tomato moment, because you'll hear helicase, I say helicase. It's just how I was taught, but a lot of times you'll hear helicase. Anyway, tomato, right? I think that's what I've said before. So with helicase, we have unwinding and unzipping. Okay, so this is now where that DNA strand is no longer kind of wound. We start to, again, we break the hydrogen bonds that are keeping everything together. We break those bonds. Okay, we break those hydrogen bonds, and that's the unzipping portion. Okay, and the unwinding is, again, we're taking these two strands that are super coiled like this. We're using the gyrase to kind of relieve the tension a little bit, and then with our helicase, we then start to unwind them. Okay, all right. Oh, I dropped the marker, hold on. Okay, it rolled over pretty far. All right, so then we have what I just lumped together, single-stranded binding proteins. So I will write that out. And these are single-stranded, oops, I wrote stranding. How about that? Woo! Single-stranded DNA binding proteins. Okay, sorry. So it's DNA, single-stranding DNA binding proteins. Okay, and that's actually what keeps things apart. I'll show you this in a little bit. I'll have to erase everything, and so I can show you all these together. Okay, so single-stranded binding proteins. We then have primase. This is going to put down the RNA primer during our DNA replication. You have, I hope you can see this. Let's see. Yeah, I'm not too low. Okay. So DNA polymerase. We have DNA polymerase 1 and 3. There are other DNA polymerases, 1, 2, 3, 4, 5, but specifically we're going to focus on these main enzymes here. So DNA poly 1 and poly 3. Then we have ligase. Okay. And oh good, you can see that because this is the last. These are the last two. And then exonuclease. That one might be a little new. So exonuclease. This is actually going to remove the RNA primer. Okay, so this removes the primase. Okay, so these are the enzymes. If you didn't get these down, or you had trouble watching the PowerPoints or the PowerPoint or watching the videos I may have linked, take a minute, jot these down. Okay. All right. So this goes back a little bit to, how do I want to do this? Oh, that's awful. Sorry. That's not much better, but this is our sugar. Okay, if I put my OH here and my H there, do you remember what sugar that is? If you said deoxyribose, then you would be correct. So remember, it's important here with sugar to know or be able to know your carbons. This is 1, carbon 2, 3, 4, and then we have carbon 5 that comes off. Okay, so that's where we get our names, carbon 3 and carbon 5. When we start talking about 5' to 3', that's what we're referring to. Okay, all right, so we have that. We are connected. I'm just going to do this. Technically, it's up. Oh, sorry, my finger really went on that one. Connected to a base. Okay, and my 5' over here is connected to, let's see, now technically this is just one phosphate group, right? So in your nucleotides, I know we've been drawing them as bubbles, circles. Technically there are 3 of these kind of phosphate groups together. So we really could continue on and do this and then continue to our 3rd. Okay, so we've just never had the opportunity to draw all that, but that's what technically there are. So there's 3 phosphate groups attached to our base, our sugar, which in this case is deoxyribose because I'm missing an oxygen here, connected to the base. All right, so we have the basic structure of nucleotides. And we're going to move on to replication. All right, so during replication, remember we have our DNA is nicely packed. It's super, super coiled, and it's nicely packed in our nucleus of the cell, okay? But there are times when we need to make more of these. So what's going to happen is we're going to take this, and I'm going to kind of illustrate it, right? It's still kind of coiled here. We kind of uncoil, and then we unwind. So if you're drawing this, definitely use actually another color. I'm going to use, just because I like it and I have it, I'm going to use hot pink. So draw my little pink ones here. Remember, this is, and I should have coiled this more. Actually, let me coil these even more. Okay, so I should have done more coiling. Okay, so remember, these are histones. These guys right here in the pink, those are histones. Those are what your DNA wraps around, again, in order to take this huge molecule, which again we know is microscopic. We already know that. But to take the huge one and condense it. It's like taking that strand of hair, twirling until it starts to wind around, and then it, you know, kind of kinks on itself. Well, in this case, we're winding it around histones, these proteins. This guy right here is the one I wanted to emphasize, and this is our nucleosome. Okay, so we have our histones, and then we have a group of histones that are, again, compacted together, and that's our nucleosome. Okay.